talk a little bit about how cells divide. And there are two different processes for cell division, meiosis and mitosis. So both of them are forms of cell division. Meiosis is used by cells to replicate gametes. So sperm cells and egg cells, this is the process they use to replicate themselves. Mitosis is used by regular body cells to replicate themselves. Meiosis creates 4 haploid cells, and each of those cells only have one copy of each gene. The first step of meiosis that we're going to talk about is prophase 1. And so here are just some examples, some diagrams of what happens during prophase 1. And basically the chromatin is going to start to condense into chromosomes. And so here are some examples of that. Remember that our X's represent our chromosomes. In metaphase 1, these chromosomes start to line up along the middle. So one way to remember this is that metaphase starts with an M and middle starts with an M. So they start to kind of meet in the middle. And then spindle fibers are also going to attach to each pair of chromosomes. And I'll show you a diagram of that in a minute. Anaphase 1, these tetrads are beginning to be pulled to opposite sides of the cell by things called spindle fibers. So let's look at this a little bit more closely. Here is metaphase 1. We have our chromosomes, and then they're meeting in the middle here. They're all lined up. Then in anaphase 1, they start to get pulled apart. So if you think about anaphase starts with an A and apart starts with an A, anaphase they start to get pulled away or apart from one another. In telophase 1, the cells divide into 2 separate daughter cells. So here's what that would look like. We have a cell here and we have a cell here. And then basically it goes through the entire process again. So in prophase 2, the chromosomes start to condense. This time they don't replicate. That's something they do in prophase 1, but not in prophase 2. So here, these chromosomes, they're starting to condense. Then in metaphase 2, again, like metaphase 1, the chromosomes start to line up in the middle. And then in anaphase, the chromatids starts to get pulled apart to opposite sides of the cell. Same thing that happens in anaphase 1. Here's just another picture of that, except we have the 2 cells this time. So the chromosomes are lined up in the middle here, and then they start to get pulled apart over here. In telophase 2, the chromosomes travel to a new nuclei. So the cells split one more time, and this time we have 4 new cells, and they each only have 1 copy of the gene. So here again, now there are 4, and each of those 4 cells has the nucleus with their chromosomes. Mitosis then produces 2 diploid cells. And so here's just a quick example of what happens in mitosis, and we're going to go through it though. So you can take a look at this picture of the prophase, metaphase, anaphase, telophase. Here's another example. See how we have metaphase, they're lining up in the middle, anaphase, they're being pulled apart, telophase. So in mitosis, we have interphase, and this actually happens before mitosis begins. The chromosomes during this phase are copied. At first they appear kind of like thread-like coils, and then they copy themselves, and then they turn into chromatids. Then, so this is an example of interphase. This is an animal cell and a plant cell. Then we have prophase, which is actually going to be the first step that we talk about. This is when the cell begins to divide. So here we have our sister chromatids that have formed. We have our spindle fibers and our centrioles at the end. This is what prophase looks like in an animal cell and in a plant cell. Metaphase is going to be the second step in mitosis. This is again where they're all going to line up in the middle. If you can look at the animal cell, you'll notice that the chromosomes are lined up in the middle here, and we have our spindle fibers to the sides. And with the plant cell, our chromosomes are again lined up in the middle. In anaphase, they start to begin to be pulled apart. So the chromatids are being separated. In the animal cell, you can see that they're moving more towards the sides of the cell, and the same thing with the plant cell. Telophase is when 2 new nuclei are formed, and then the chromosomes start to go back to more thread-like rather than being rod-like, and this is the end of mitosis. And so here you can see that now we have the 2 new plant cells, and we have the 2 new animal cells. Cytokinesis occurs after mitosis. This is when the cell membrane moves inward and you finally have the creation of the two completely separate cells. And they are identical. They have identical chromosomes in them. So again, just a quick review. Take a minute to kind of go through this and look at what happens in the different phases of animal cell mitosis. Another example of the cell cycle, you can go ahead and kind of look at those, try to better understand what's going on there. And then take a minute and see if you can fill in the information that was supposed to be here. If you try to fill it in, and then go back and check your answers if you need to. There's an animation on this page that'll kind of give you an idea of what's going on with mitosis, and we'll look at this in class too. This is the exact same thing, it's just been dyed a little bit different color so we can see what's going on a little better in the cell. Another example of what's going on, another way to look at it. It's the exact same thing, just with some different colors. Maybe it'll stick with you better. These are some of the references that I used for this presentation. I know that some of this terminology is a little bit confusing. We're going to be making models of all the cells in mitosis and all the stages in meiosis. We're also going to look at some pre-made models. This is something we're going to do some interactives online with as well. So again, if you're feeling confused, please ask me. And don't forget to do your notes review.